Hello, students. Let's continue our first immunology lecture. Pathogenicity and virulence. And this material was presented in the first part of the lecture. It's about virulence of microorganisms, virulence factors, including toxins, exo and endotoxins. And our second part is about immunity, the definition, types of immunity, and factors of non-specific resistance or innate immunity of human. And what is immunity? It's the protection from living bodies and substances that possess the alien genetic information. It's the set of biological mechanisms aimed at maintaining the stability of internal environment and protecting the human body from infectious and other alien agents. Types of immunity are listed at this slide, and this material is not very difficult for independent work, so please find the information about the definitions of these types of immunity and write down in your copybooks. And this task was estimated in practical classes. And the immunity divided into two main mechanisms. It's innate immunity or non-specific resistant and adaptive immunity. Adaptive immunity we will study later, and now we start with non-specific resistance. Why it's named non-specific? Because it's the same types of defense mechanism against any pathogen and also against physical and chemical damaging agents. These mechanisms can be non-inflammatory, which acts on the surface of skin and mucous membranes, and other mechanisms, um, inflammation, which acts in tissues and the bloodstream. And what is the main properties of innate immunity? It's genetically older than adaptive. It's the first line of defense, and it exists before and reacts immediately after contact with pathogen. And it's not the development of immunologic memory in innate immunity. It's a property only of adaptive immunity. And mechanisms of innate immunity stimulate and shape adaptive, so they work together. And innate immunity includes anatomical barriers with some mechanical, chemical, and biological factors in it, the humoral components present in the serum, like complement, coagulation system, cytokines, interferons, and cellular components. It's a phagocytic cells like neutrophils, monocytes, macrophages, and also natural killer cells and eosinophils. Here you see the barriers, phagocytic cells, non-phagocytic but very important natural killer cells, and humoral components like complement system. And we start with barrier function of skin. Intact skin is impermeable for any microorganisms due to their mechanical strength. The special proteins like keratin and collagen present in it, keratin in the epithelial cell at the surface and collagen in the deeper layer of skin, and they're very strong. Other defense mechanism is the desquamation of epithelial cell. Bacteria can stick to this cells, but they eventually die and mm, removed from the surface of skin with all bacteria and viruses in it. The sweat glands produce lactic acids and enzymes which have antibacterial properties. And unsaturated fatty acids in the secretion of sebaceous glands also suppress the multiplication of microorganisms. Barrier function of mucous membranes is also very important. And here you see the micrograph of ciliated epithelium. It's very important mechanism of defense in respiratory tract due to mucociliary elevator. This cilia moves and swept out all bacteria and viruses entering the respiratory tract. In gastrointestinal tract, the epithelium is non-ciliated, but the mechanical factors also present in it. It's the peristaltic of intestine. Important chemical factors in mucosa. It's acids mostly in stomach, which prevent the multiplication of almost all bacteria. In the tears and saliva, the antimicrobial substances like lysozyme and phospholipase A present. In respiratory and gastrointestinal tract, special substances, defensins, possess the antimicrobial activity, and in lungs, the surfactants present, they act as opsonins, so they make easy the phagocytosis process. And very important biological factor in mucosa, it's normal microbiota, which produce some antimicrobial substances like bacteriocides and peroxides, and it compete for nutrients and colonization with pathogenic bacteria. And here you see the action of microbiome as a defense factor. The microbiome resident bacteria normally colonize all surfaces of non-sterile cavities in human body, and if some pathogens invade the site, they cannot attach to the epithelial cell wound surface, and they easily excluded from this microbiome. But if normal microflora is damaged, the disease develops due to excessive multiplication of pathogenic microorganisms. And the next question is very important for stomatology students. It's the protective properties of saliva. It's also one of the mechanisms of non-specific resistance. Maybe you know that salivary glands produce 1/2 to 2 liters of saliva every day, and the saliva provides a mechanical cleaning of the surface of oral cavity and contains many antimicrobial substances. If production of saliva is not enough, the risk of dental diseases increase. Like in this patient, you see the caries in many teeth. And which antimicrobial substances exactly present in saliva? They are lactoferrin, it's iron-containing transport protein, it's bacteriostatic because it competes with bacteria for iron. It interferes with bacterial adhesion and kills bacteria through depolarization of cytoplasmic membrane. The Lactoseperoxidase, it's a protective enzyme. It provides bactericidal action in combination with thiocyanate or in the peroxide and prevents the adhesion of streptococci. Streptococci, which present in oral cavity of all people. Histatins, the substance which kill the candida fungi and prevents the co-aggregation of bacteria. This process of co-aggregation. Cystatins are inhibitors of bacterial proteases and they regulate the inflammation. Protein chromogranin A is produced by some salivary glands and has antibacterial and antifungal action. And the secretory leukocyte protease inhibitor, very important factor of defense, it inhibits the infectivity of HIV virus and also possess bactericidal and fungicidal activity. But the most important bactericidal factor of saliva is the lysozyme. It's the enzyme that destroys bacterial cell wall. It acts as a moramidase. This is a structure of the lysosome, its active center. And do you remember the composition of bacterial cell wall, peptidoglycan? It's composed of long polysaccharide chains and short polypeptide bridges. And the lysosome cleaves these links between N-acetylmuramic acid and N-acetylglucosamine. Lysosome has a bactericidal and bacteriostatic effect on different type of microorganisms, especially against gram-positive. And also it stimulates phagocytosis and antibody synthesis. Lysosome also produced by macrophages and neutrophils, can be found in cartilage tissues, and it's present in nasopharyngeal mucus and tears and milk and urine and many biological fluids. And also it's used as a medicine for treatment of inflammatory process, including especially in oral cavity and upper respiratory tract. It produces like sublingual pills, and it's rather effective, especially if infection caused with gram-positive microorganisms. And here see the main mechanisms which present in oral cavity. It's production of antimicrobial peptides listed below, and the lysosome production, the Lactoferrin, Lactoseperoxidase, and the resident flora. This microbiome is very important as a defense factor, and the mucin produced with goblet cells. It's a surfaces of mucosa in oral cavity. In tissues, the other factors of innate immunity present is the macrophages and complement system and cytokines and platelets. And in this diagram, some factors of adaptive immunity are also shown. It's the T cells and plasma cell and production of immunoglobulins. It's not part of innate immunity, but it's also present in oral cavity as defense mechanism. If bacteria or virus overcome the barrier of skin or mucosa and enter bloodstream or tissues, the next line of defense is some cellular components of innate immunity, mostly the neutrophils, which have phagocytic properties and participate in inflammation, macrophages, which are also phagocytes, and they're able also for extracellular killing or infected or altered target cell. They participate in tissue repair, and the very important function of macrophages is the antigen presentation for specific immune response, but we will study it a bit later. Natural killer cells can kill viral infected or malignant cells, altered cells, and eosinophils, important in protection against certain parasites. But the most important mechanism, cellular mechanism of non-specific resistance, is a phagocytosis. In phagocytosis, specialized cell engulf live or killed microorganisms and digest them by intracellular enzymes. The most important phagocytic cells are the macrophages, the polymorphonuclear leukocytes and blood neutrophils, and macrophages, which present in connective tissues and alveoli, in the vascular epithelium, in neural glia, in Kupffer cell, in liver, and other. And phagocytes may be found in oral cavity, and they migrate there from the gingival pockets. And here you see the process of phagocytosis in one diagram, and it includes some stages which we'll study separately. The first stage of phagocytosis is the chemotaxis. It's the movement of phagocytic cells to the invading pathogens. The chemotaxis is activated with fragments of complement system, globulins, lymphokines, degradation products of fibrin, collagen, and cells. The second step is the adhesion. Due to interaction between lectin proteins, which is at the surface of phagocytic cell, and some sugars at the surface of bacterial cell. And adhesion is enhanced with opsonins, which may sort of antibodies or component C3 from complement system. The third stage is endocytosis. It's membrane activation and appearance of pseudopodia, which engulf the foreign cell. In this slide, it's erythrocytes, but the bacteria engulf it in the same way. And the phagosome formation appear. And remember that hydrophobic microorganisms ingest easily than hydrophilic. And then plasma granules fuse with phagosomes and inject their content. And these plasma granules mostly contain enzymes and some bactericidal substances. The fourth step is the intracellular ingestion. The digestion of foreign cell can be proceed in two ways: oxygen dependent, with participation of bactericidal substances like superperoxide ion, hydrogen peroxide, hydroxyl radicals, oxygen dependent, and the oxygen independent way when cationic proteins, lysozyme, lactoferrin destroys the bacterial membrane. And after digestion, small molecules are released from phagocytes, but large molecules can remain inside phagosomes. But sometimes phagocytosis may be incomplete, and instead intracellular digestion, we observe intracellular multiplication of pathogens. Because you know from first part of this lecture that many bacteria have antiphagocytic structures or substances in their composition. Like gonococci and pneumococci have a capsula which protects them from digestion. The Mycobacterium tuberculosis contains a high concentration of lipids in its cell wall, and it inhibits the fusion of phagosomes and lysosomes. Bacteria like Legionella can inactivate lysosomal enzymes, and small intracellular parasites, Rickettsia, can come out of the phagosomes and multiply in cytoplasm. And here you see gonococci inside phagocytic cell, and this is multiplication of mycobacteria inside macrophages. And here is Rickettsia multiplying in cytoplasm. Do you see that many bacteria can survive inside phagocytic cell, multiplying it, and we see that these bacteria possess the antiphagocytic and aggressive properties. Phagocytosis is a cellular defense component of non-specific resistance, and some humoral factors are also present in our body. It's bactericidal substances of serum, and they're listed in this slide. And this also piece of your independent work. Please find which effect has all these defense factors. It's not difficult to find in any resources. But the most important humoral factor of non-specific resistance is the complement system. It's a complex of serum proteins. It contains 9 factors, which forms a cascade system. Normally, the complement components are inactive, but they activate when pathogen enter the human body. There are 2 main pathways of complement activation. They named classical and alternative. Classical pathway is possible only in immune organism when antibody is present, and they react with microbial antigen, and this complex antigen antibody triggers the activation of complement system. The first fraction, C1, attached to it, it undergoes the splitting into two parts, and one of the enzyme which activates the next component, C2, then C4, C3, and C3 component is a key point for classical and alternative pathway. Alternative pathway possible only in infection caused with gram-negative bacteria, but it doesn't require the participation of antibodies, so it's possible in non-immune organism. And it's a bit shorter, starts with C3 component. And then terminal fractions from 5 to 9 bind together and form the membrane attacking complex, the large pores in the cellular membrane, and cause the lysis of target cell, bacterial cell, or any foreign cell. Upon the activation of complement, some active subfractions are released. These fractions, C3b and C4a, have properties of opsonins, so they activate phagocytosis. These fractions, C3a and C5a, activate the respiration of phagocytes and the oxygen-dependent way of killing of bacteria in them. And also they participate in allergic reactions. And the component C5b attracts the neutrophils and cause the dilatation of blood vessels and increasing of their permeability. You understand that activation of complement system leads to local inflammatory process. When complement works, we can observe the redness, the swelling, and it is painful, all signs of inflammation. And here is more details about complement activation in classic and alternative pathway, the cascade reaction, which results in formation of pores in the membrane of target cell, bacterial cell, and cause the lysis of it. And you see the bacteria before the complement activation and after the cell lysis and cell death. And the complement fixation or complement activation starts with formation of complex antigen and antibody, and all 9 fractions must gather together to kill this bacteria. This mechanism forms the membrane attacking complex and release all cytoplasm outside the cell. The complement system is mostly effective against bacteria, but we have also antiviral factor of protection. It's the interferons. They inhibit viral reproduction and also growth of cancer cell, bacteria, and sometimes protozoa. They act mostly in the early phase of viral disease and limit the spread of viruses. They induce the synthesis of cell-encoded proteins that inhibit translation of viral matrix RNA without affecting the translation of cellular matrix RNA. There are 2 groups of interferon in human body. It's the alpha or leukocytic, beta or fibroblast, and gamma or immune interferons. Interferons are used like a medicine for prevention and treatment of viral diseases, maybe human donor interferon, or also we have genetic engineering interferons. And it's an effective measure to stop the infection like influenza in the early stage. And how does it work, this interferon? What's the mechanism of action? Virus, mostly RNA genome virus, invade human cell and start to multiply in it, but this infected cell produce special protein, interferon, and release it into intracellular space. And the healthy cell, non-infected, acquire this interferon, transport inside, and interferon activates some genes in its nucleus to produce antiviral protein. And if this virus tries to multiply in the cell, these proteins inhibit the production of its enzyme, and the viral infection is stopped. So when we use the interferon as a medicine, mostly in nasal drops, it protects our cells from multiplication of viruses. And that's all I want to tell you about innate mechanism of defense. If you have any questions, contact me by email, and thank you for your attention.